Okay, good afternoon, everybody. And this afternoon, we're actually going to be discussing 3 topics. Very briefly, we'll finish off our discussion about phylogenetics. And we'll end that by talking about the tree of life, which lets us segue into our next discussion, our first group of organisms that we are going to be discussing in the semester. And those are the viruses and the prokaryotes, although the viruses are not really organisms, as we'll hear. Okay, so we learned in discussing phylogenetics that, in fact, these phylogenetic trees have many applications, not only in systematics and taxonomy, that sort of thing, but practical application as well. We saw, for example, how unknown whale meat could be identified by comparing DNA sequence to known whale sequence, so that the blocks of unknown meat could be identified as being specific whale species. So that is one typically practical way in which this phylogenetic could be used. But the phylogenetic trees can also be used to hypothesize about ancestral traits present in groups even which are not extant. What I mean by that is we can use what we know about groups that are extant, that are still living, to make inferences about other groups who share their ancestry, even if they're extinct. This is called phylogenetic bracketing. And for example, it's most easily explained. If I have a look at this phylogenetic tree, and this is a phylogenetic tree, is an extremely interesting phylogenetic tree because it is kind of a grouping of what we might associate with the reptiles. And so we take as an outgroup, here are the lizards and the snakes, but we are particularly interested in this group here. The crocodiles, birds, the theropod dinosaurs, birds we strongly suspect have evolved from theropod dinosaurs. Here are theropod dinosaurs in the fossil record, other than the birds, and then other dinosaurs. We believe that this entire group arose from a common ancestor, and that derived traits arose in this common ancestor, which have been inherited by all of these groups here. They have not been inherited by lizards and snakes. Let's look at the extant groups, the groups that are still living, the crocodiles, the crocodilians, and the birds. They share an enormous number of traits, believe it or not, even though they look so completely different. They brood their young. Did you know that crocodiles brood their young? Well, they do. They dig a nest, lay eggs in it, and they care for that nest. They actually will regulate the temperature in the nest. They will scrape sand off or pile sand on top to regulate the temperature. They guard the nest, and when the young hatch, they guide the hatchlings to water. They communicate with their young by chirruping and by singing. And the singing, the noise they make, is derived from exactly the same anatomical structures as are used in the birds. It's an area of the thorax, of the throat rather, called the syrinx. And the birds use that to make their song. Birds, of course, do exactly the same thing. They lay eggs in a nest, which they care for. They brood their young and they care for their young. So here there are these 2 groups. By the way, they also share anatomical features. They share a 4-chambered heart and various other things as well. They are not nearly as dissimilar as we might think. Morphologically, they are very different to one another. But we believe that those features that I've mentioned are shared because they've been inherited from a common ancestor. They are derived traits that are present in these 2 diverse groups. But we know also that the birds are strongly allied to the theropod dinosaurs. Here they are actually a sister group to the theropod dinosaurs other than the birds. And that grouping there is close also to the other dinosaurs. We can bracket the features that we know about, and we can suggest that these groups especially the theropod dinosaurs, and probably the other dinosaurs as well, shared these ancestral traits. They shared these ancestral traits because this is where they arose, where they were derived from. Okay, those traits are not shown by the lizards and the snakes, but we can infer that they were shown by these, which are now all extinct, and we can hypothesize about their lifestyle. And this is borne out when we look at the fossil record. For example, for the theropod dinosaurs, we find very strong evidence that they laid eggs in a nest, which they cared for and which they actually brooded. There are fossils of parents which were killed while they were busy brooding a nest. So that is very strong evidence that this is in fact true, that these groups all share those features, even though we can't hear them sing, and we have no fossils of their hearts. We can make a supposition that they did in fact use song for communication, and that they had four-chambered hearts. So the use of phylogenetics was, even predating DNA sequencing and everything, was used to begin to try to construct what biologists colloquially refer to as the tree of life. Some sort of a phylogenetic tree which links all living things together. We saw at the very start of our discussion how Darwin at least began to work this out in his brain, did thought experiments about how this might work. That tree of life has adopted many different forms as more and more information has been acquired about more and more different organisms, and biologists have tried to link them all together. But it has been particularly well informed by DNA sequencing, because DNA records evolutionary history extremely well. And it allows us to draw conclusions about relationships which are really not necessarily apparent if we were just to use morphology. Organisms can look completely different to one another, and yet share a huge amount of DNA homology, suggesting that they do in fact belong together. Likewise, it has to be said, some groups which have formerly been put together because they looked similar are revealed to be only distantly related once we look at their DNA sequence. So the structure of the tree of life has changed tremendously over time. Originally, biologists basically grouped organisms into two major groups, which are the plants and the animals. There were those things which were photosynthetic and those things which were heterotrophic. With the discovery of the whole microscopic world, some attempt had to be made to place those organisms into some sort of structure. And ultimately, by the 1960s, most biologists recognized five giant kingdoms of living things. And these were the Mrna, these were the prokaryotes. And it didn't matter what the prokaryotes did. It didn't matter whether they were photosynthetic or heterotrophic or whatever. If they had no nucleus and just a cell membrane, then they were put into the Mrna. The other group that was recognized were the protists. Now, the protists actually continue to be recognized as a group until really quite recently. And you'll still, I think even in your textbook, there may be reference to the protists. This was a group which was a mishmash largely of single-celled organisms, all eukaryotic. And the only thing that put them into this kingdom was the fact that they were neither plants, nor were they animals, nor were they fungi. And so this came to be a classic polyphyletic group. That is, it included erroneously, but really for the practical purposes, it included a whole range of very diverse, really unrelated organisms, which were just put together into this artificial grouping because they were none of these. That is no longer the case. And this grouping is not recognized as being valid. And instead, these are included, probably correctly, into other groupings. And this has been especially well supported by DNA evidence. So now the most recent groupings is to recognize 3 major domains. And these are the domain Eukarya, the domain Archaea, and the domain Bacteria. And the domain, if we go through the domain Eukarya, we'll find in each of these, if we look at these different groupings here, you'll find in these groupings members who were formerly placed into the protists. The protists have all been separated out and based not only on biochemical things like do they photosynthesize, etc., but on their DNA sequence, they have been placed together with many of the other eukaryotic groups, both plants and animals. And you'll see that scattered throughout here, for example, the feature of photosynthesis occurs in several different places. It's not necessarily a defining feature for a particular group. You will notice that if you look carefully at this, that there are some very obvious groupings. For example, the plants and the green algae are very closely grouped together. That was not always the case. Many of the green algae were placed in the protists, but in this scheme, they're actually extremely close to the plants, whether or not they are multicellular or unicellular. They perform plant photosynthesis and have chloroplasts, which are extremely similar. The other one that might surprise you a little bit is to see that the fungi and the animals are actually sister taxa. They're actually relatively closely related to one another, both obviously heterotrophic, and there are many DNA similarities as well. So this is the domain Eukarya. It includes all of the prokaryotes. I mean, all of the eukaryotes, I'm sorry, all of the eukaryotes. There is something to notice, however, and that is, okay, here we have these groups. These groups appear to be quite clear. They seem to be a true clade there and a true clade there, true clade there, etc., etc. But have a look at the ultimate ancestry, and you'll see that here there is a multiple branch. If this was one of those trees which didn't have the right angles, instead had an angle, you'd see it like that. Okay, this is called a polytomy, and it infers that we don't know. We do not understand this ancestry. We do not understand. We know that these are related, but we cannot say how they are related. We simply know that these are related and that they are further down the line, they're related to these other groups. But here, what the ancestry which led to this particular disposition of the clades is unclear to us. So the next domain is the domain Archaea. And it is, here's the domain Archaea. What is characteristic of the domain Archaea? They are prokaryotes. They are prokaryotes. They are strange organisms, many of them, that perform some strange biochemical processes like making methane, etc. Often live in extreme habitats and this kind of thing. But they are true prokaryotes. They only have a cell membrane, and they in fact are also prokaryotes. The other prokaryotes, of course, are the both bacteria, and they are in their own domain, the domain bacteria. We might, just off the top of our heads, think that the domain Archaea and the domain of bacteria would be closely related, that they might even be sister taxa, but they are not. They are not. In actual fact, the domain Archaea are more closely related to the eukaryotes than they are to the bacteria. There is more similarity between the Archaea and the eukaryotes than there is between the Archaea and the bacteria. This is a really strange thing, but it is true. And in fact, we believe that the eukaryote cell, with all of its organelles that arose from endosymbiosis, etc., these were actually evolutionary steps using archaeal ancestry to lead to these two groups, the Eukarya and the Archaea. The bacteria proceeded along an entirely different line. Here is the common ancestor for all life. There was some major division into two populations, one leading to the Archaea and the Eukarya, one leading to the domain bacteria here. You can see the bacteria proceeded along there. Again, however, there is a polytomy. We do not fully understand the ancestry of these very clearly distinguished bacterial groups. So that is the domain system. Three major domains with their various branches. Okay, so with that, let's proceed to talk a little bit about viruses first of all, and then about bacteria and the Archaea. We'll have very little to say about the Archaea, unfortunately, but some more to say about the bacteria. Okay, viruses obviously of tremendous interest to us at the moment, since the entire human population is enthralled now in trying to fight a great pandemic. And what we're seeing is typical viral behavior, typical viral production of disease, and in a very, as always, for viruses, in a very restricted host population. And viruses, first of all, we need to state right up front, viruses are not cells. They're not true organisms. They are not based on a cell, which is the basis of all life on the planet. They are not alive. They have no metabolism. They are completely unable to reproduce themselves. They have to co-opt the machinery of a host cell in order to reproduce. They are an infectious particle consisting, at their simplest, consisting of a set of genes, a genome, which is packaged into a protein coat. There may be elaborations on that structure, as you will see in a minute, but that is the simplest virus, a genome enclosed in a protein coat. They're very much smaller even than prokaryotic cells, and they're much simpler in structure in that they don't have a cytoplasm, they don't have any cytoplasmic elements, nothing like that. They are obligate parasites, so they always cause disease in whatever they always have the potential for causing disease in whatever host they occur. And they have a very, very wide range of different hosts, all the way from bacteria, all the way up eukaryotes, all of the eukaryotes. Virtually every living thing has its own identified viruses that will infect it. They cannot reproduce anything. They can't do anything without co-opting the host machinery to do it for them. Now, the capsid, the protein coat around the genome is called the capsid. And the capsid may also sometimes include some enzymes which are derived from the host, coded by the viral genome. But they may, in some viruses, they merge with a few enzymes of their own. So they are really in this kind of gray area between life and non-life. And it's very difficult to say how they arose. They probably arose originally as mobile genetic elements, as particles of something else's genome that was able to move from one organism to another. So the way in which viruses reproduce, the raison d'etre is to manufacture more virus. They are required to enter a host cell and to co-opt the machinery of the host cell to turn it into kind of a viral factory. So this is the very first stage of all viral infectivity, is that they have to attach. The viruses have a narrow host range. Some have wider range, host range than others. The current SARS-CoV-2 or COVID-19 virus appears only able to infect mammals. And we only know of a few mammals that it does infect. We know that it can infect other primates. We know it can infect cats, dogs, and bats and things like that. But it appears to have its major pathogenic effect appears in the human population. So viruses, first of all, they have to be able to recognize the host cell that they can profitably enter to get it to produce more viruses. To do so, they have a recognition mechanisms. Those are usually in the outside covering of the virus. And they consist usually of proteins which will bind to specific proteins on the host membrane. Once they bind, they get entry into the cell, and they eject their genome into the cytoplasm of the cell. They get the host then to replicate both the viral genome and to produce all of the viral proteins that would make up a new viral particle. Those, though they use the normal mechanisms of the host cell to do so. And once they have manufactured all of the viral proteins and the new viral genomes, many copies of the new viral genomes, these classically will self-assemble. Once the viral protein bits approach one another, they will automatically assemble into new viral particles, enclosing the new viral genomes, and they then leave the host cell. There are two ways to leave the host cell. The one is that often many viruses will actually burst or lyse the host cell once viral reproduction is complete. In others, the cell will actually continue living for quite some time, producing viral particles, until eventually the machinery of the cell is so co-opted, so damaged by the presence of the virus that the cell dies. So first of all, just remember, at its simplest, we have a protein coat which encloses a genome. Now, the other characteristic of living things on the planet is all life is based on the use of DNA as the kind of master molecule. Remember the central dogma that DNA is transcribed to messenger RNA, which is translated to protein. So that is the central feature of all living things on the planet. However, in viruses, you do not necessarily have DNA. It may also be RNA. We divide the virus, we see viruses that have DNA, and we see viruses that have RNA as their genome. Not only that, they're really weird, because in some viruses, the DNA, in some DNA viruses, the genome is a linear. It's a long strand. In others, it is circular. Not only that, but in the DNA sometimes occur in some viruses is single-stranded. It's very, very strange. It looks almost like RNA, excepting that it is DNA. It has all the characteristics of DNA. It has thymine, not uracil, et cetera, et cetera, et cetera. So just immediately look what we've looked at the diversity we've got. We've got some are DNA, some are RNA, some are linear, some are circular, and some are single-stranded and some are double-stranded. Same thing applies to the RNA viruses. Some of them is linear, some of them it's circular, some of them is single-stranded, and some of them it's double-stranded. They are incredibly diverse. Their genome structure is very diverse. By the way, although don't worry about the names here. You don't have to remember that, but this is one of the principal ways that we use for classifying viruses, which is very, very difficult to do. But these are some of the principal ways in which viruses are classified by the structure of their genome. Physically, there are many elaborations on that simple structure that I told you about, the genome enclosed by a capsid. Here, for example, this is in fact one of the very first viruses that was ever identified and visualized. This is tobacco mosaic virus, and it is a typical helical virus. And this, what you see, it's an RNA virus. Here's a strand of RNA there. And it is enclosed in protein units, each of which is called a capsomere. And the capsomeres go together to form the capsid. And in these, it forms a helical structure like this, with the genome wound around inside there. Now that's important because this is the basis of the capsid structure of COVID, for example. But it's also the structure of some other famous viruses. Ebola virus, for example, has the same helical structure. This here is a much simpler structured virus because this one, all it has is the capsid on the outside, and it's called an icosahedral virus. And here on the inside is the genome there. And on the outside, there's this geometric crystalline structure formed from the capsomeres that link together to form the capsid. These spikes sticking out here are the recognition spikes that it would recognize its host cells with. This is typical of an adenovirus. Adenoviruses are common viruses in human beings, and one of our common colds, for example, is produced by an adenovirus. This is much more typical for many of the animal viruses. This is a virus which has an outer covering, which is an envelope. The capsid structure is on the inside here. And in this case, this is influenza virus. And you can see here, it has these helical structures here, but the genome in influenza, uniquely, the genome is cut up into 8 pieces, each with their own capsid covering on the inside here. But the whole lot are enclosed in an envelope, and that envelope is derived from host membrane. It can be endoplasmic reticulum. In other viruses, it's the Golgi apparatus. In other viruses, it's the outer cell membrane that this membrane is derived from. But it has a particular structure which is really important, which is that this shell is derived from a phospholipid structure, because that's the structure of the cell membrane. But embedded in it are viral proteins. And those viral proteins are governing the recognition and attachment to specific host cells. So this is electron micrograph. You can see the envelope here, the double membrane there. All of these lumps there are the protein spikes. And then on the inside here would be the capsid with the genome, enclosing the genome. So now I told you, viruses infect every single living thing. In actual fact, there's a virus that infects other viruses, believe it or not. But every living thing has its own sets of viruses. And this very complex virus here is in fact termed a complex virus, is a virus that infects bacteria. So this is a virus that infects bacteria is called a bacteriophage. And this is the most extraordinary structure. I won't go into all of the details of it, but it's a little injection syringe. Here is here at the top is the capsid. It contains the genome. In this case, this is a DNA virus, and it's containing DNA. And all of the rest of this here is an elaborate injection syringe. And it sticks to the outside of a bacterium, and it sticks a tube right into the cytoplasm of the cell, and it injects the DNA down that tube. So that's a bacteriophage. So this here is one of the most infectious of all of the human viruses, and this is the measles virus. Its infectivity is about 3 times or 4 times the infectivity of the Omicron virus that is resulted in the current wave of COVID. So I repeat a really important point about the viruses. They tend to have a narrow host range. And even within the host, within a specific host, they only infect specific cells. And if they're not able to gain entrance to those cells, the viral infection cannot proceed. So they have a limited range that they operate within. So they're in the bacteria, in the bacteriophages, we see the following stages of infection. And they're important to understand because they actually have reference to the way in which viruses like the measles virus cause disease in humans as well. So here's the bacteriophage that we saw before. And this is a typical simple life cycle. First of all, the bacteriophage injects its DNA, in this case, into the cytoplasm. The host DNA is actually, in this case, is degraded. And all of the machinery of the host cell, all of the transcription machinery and the translation machinery and everything is co-opted to read off, first of all, copies of the genome of the virus, and then to read off masses of the proteins, which are going to make the viral particle. The viral particle, once all of those proteins are present and mature, they will automatically assemble, and they will package the DNA and then burst the bacterial cell and escape. And they obviously kill the cell, they lyse it. So this is called a lytic cycle. And for many viruses, this is the typical cycle. But there is in bacteriophages, there is another way in which the viruses can interact with the host. And that is that they can become integrated with the host genome. So here's the lytic cycle that we just seen. Some bacteriophages don't necessarily go straight into the lytic cycle. Instead, what they do is this little blue circle here is the DNA genome, right? Instead, what they do is they integrate the viral genome into the host genome. Here's the host circular chromosome. And every time the cell, this bacterial cell divides, it replicates the viral DNA as well. So this is called a prophage. And it has no effect on the cell. It has no deleterious effect on the bacterial cell. But you're going to realize that these bacterial cells reproduce very fast. And this is becoming spread, this prophage is becoming spread through the whole population. Then on usually an environmental cue of some sort, this prophage can leave the the integration. It can exit from integration and form a little circle of viral DNA again, and now enter the lytic cycle. So this can persist for a very, very long time. And in fact, we see certain bacteria which have portions of a lytic virus in them, which appears to become completely integrated into the bacterium and never result in lysis. But in others, they alternate between these two, between this stage here, which is called the lysogenic cycle, and this, which is called the lytic cycle. Viruses which are able to do this for a long time are called temperate viruses. Those that go straight into this lytic cycle are called virulent viruses. Now, this has some import in human and in animal viruses as well. Not exactly the same, but there are many animal viruses which are able to integrate themselves into the genome of the host and remain silent there for a very long time. And a typical example are the herpes viruses. And for example, chickenpox, which is a childhood disease caused by one of the herpes viruses, has an initial stage where it appears mostly to be lytic. It appears that it causes cells to rapidly become damaged and to die, and that is what produces the disease symptoms. But once the immune system beats back that infection of the virus actually becomes integrated into nerve cells, and it becomes integrated in exactly the same way as a prophage in the host genome. And it doesn't necessarily, but it may appear many years later as another disease, which is called shingles. So that's this, although it's not exactly the same kind of mechanism, it's at least is analogous. Okay, so I just want to tell you a little bit about a couple of our really important viruses. And the first, of course, that we have to discuss is SARS-CoV-2. And that is the cause of our current COVID epidemic. So the coronaviruses are called coronaviruses because they look like this under electron microscope. It looks like a crown there. And what you're looking at is an envelope. It's, again, this is an enveloped virus. And it is an RNA virus. It has a single strand of RNA as its genome. And this is the envelope here. And these are all viral proteins that are sticking out like knobs on the outside. These are the infamous spike proteins. Here's a diagram of one, a picture of one rather. And you can see all the spike proteins here. And these are important because this is what governs the attachment of the virus to its host cell, and it governs its entry into the host cell. It's also what our current vaccines are directed against these spikes to stop their activity, so that they can't enter the cell. So here's the envelope on the outside. Here are the viral spike proteins there. And if we look on the inside, there is a helical capsid there, and it encloses this long strand of RNA. And that RNA, when it's injected into the host cell, looks exactly like messenger RNA to the cytoplasm, to the ribosomes of the cytoplasm, and they will immediately begin to translate it. So that's its relatively simple structure. And just to tell you very briefly, I'm not going to go into enormous amount of detail, but to tell you very, very briefly about some of the features of its replication. Here's the virus here. You can see the envelope with the spike proteins, and then on the inside, here's the capsid with the RNA genome. It attaches to a specific protein, which is called the ACE2 receptor. And only certain cells of the body have ACE2 receptors. The most important ones are being cells in the respiratory tract, the lungs, the heart, and in the blood vessels. And immediately you hear that, you'll realize how it is that COVID produces some of its major symptoms, okay, which often involve clotting disorders and things like that. So when it attaches like this, the cell actually swallows the virus. It takes it in, and if you think back to your first semester, this takes it in by a process of endocytosis. A vesicle is budded to the inside containing the virus. The viral membrane then unites with the membrane of the vesicle, and it opens up and spits the genome into the cytoplasm. That genome is RNA. It looks like messenger RNA, and the host ribosomes immediately translate, and they translate for two purposes. The first is that they start to read off viral proteins. They read it off as a giant molecule, which is then cut up into smaller pieces to form the viral particles. And at the same time, they reproduce the entire genome over and over and over and over again. So that is this whole process here. What are they doing? They're producing massive copies of the viral genome, and they're producing the viral proteins. That process of translation, think back to your first semester of our discussion about how translation takes place. And you'll remember that sometimes translation takes place on endoplasmic reticulum. And this is exactly what happens. The translation takes place on endoplasmic reticulum, and it embeds proteins in both endoplasmic reticulum membrane and in Golgi body membrane as well. And the Golgi body now forms new vesicles. This is what they look like. Here is what is going to become an entire viral particle there. Here is its envelope there. Here is the genome, which has been reproduced and has been enclosed in new capsid proteins. That gets enclosed in this envelope here, and the whole thing is now enclosed in a new vesicle. And that vesicle is exported to the cell surface, and the viral particle is spat out. And this persists for some time before the cell is so weakened that it will die. But a single cell can produce thousands of these new viral particles using this mechanism. The really important things to remember are that the membranes that we see on the outside of the COVID virus are derived from endoplasmic reticulum and from the Golgi, with viral proteins embedded in those membranes, enclosing copies of the genome, which are produced in the cytoplasm during the process of replication of the viral genome. So that's SARS-CoV-2. That is a fairly typical viral scenario for an RNA virus. Most animal viruses are RNA viruses. So I just want to quickly mention one other important human virus, and that is the virus that causes the disease of AIDS, which is the human immunodeficiency virus. I'm not going to go into a lot of detail, but I just want to point out one or two things about them that are really significant and interesting. First of all, here's the virus itself. Again, you'll recognize it's enveloped. It has an envelope which is derived from host membrane. In this case, the membrane is derived from the cell membrane, from the plasma membrane. On the inside, here's the capsid. It has this conical capsid. and it has 2 strands of RNA there, and it has its own a few of its own enzymes. The most important one here is called reverse transcriptase, and I'll show you the importance of that in a second. So the human immunodeficiency virus, first of all, has a very narrow host range. It only infects and causes disease in human beings. It is strictly a human virus. It attaches in the human being, it only attaches to cells which have particular sets of proteins on their surface. And the most important of those cells are members of the immune system. In particular, a group of cells called the T helper cells. So they attach especially to T helper cells. It's another classic way of getting in. The membrane fuses with the cell membrane. This one is not taken in by endocytosis like SARS-CoV-2. Instead, it unites with the membrane and just spits the capsid in. And the capsid is broken down, and the RNA is released into the genome. Here's the importance of this viral enzyme here. Reverse transcriptase will come to the RNA, and it will transcribe not another RNA, as it would in SARS-CoV-2, but it transcribes a DNA molecule. Now, the normal process of transcription is from DNA to RNA. DNA is transcribed to messenger RNA. That is why this is called reverse transcription, because here we have RNA, which is transcribed to DNA. And that DNA is then doubled. And they now have a double strand of DNA. And what does it contain? It contains all of the information for the virus. And that now enters the nucleus and becomes integrated in the host genome as a provirus, where it may remain silent for a very, very long time. In the case of these helper T cells, it will remain silent until this cell is activated by the immune system. When this cell responds to an immune stimulus, then the viral information begins to be transcribed and translated. And once that happens, once it is activated, the cell will produce masses of viral RNA. Some of that RNA is genomic RNA, there it is there. Some of it is functional RNA, which is translated by the cell into all of the viral proteins. And the capsid then assembles inside the cytoplasm and encloses the RNA, and it is budded out. There it is, here's the capsid, and it gets budded out. That's how it acquires its envelope there. Now, I just want to mention to you that there are a lot of steps here. And some of these steps are important because they offer us chances to act therapeutically. The first place where that is possible is here, by trying to inhibit this reverse transcriptase, to try to inhibit the replication of the RNA as a DNA molecule. That is a line which is still used. It's not particularly successful, but it is used. It's a very good prophylactic. If people have been exposed to HIV, but not yet suffered a rampant infection, then this is a good way to prevent them from proceeding to a full infection. But the major way in which HIV is now treated very, very, very successfully is to inhibit this process here, the production of these viral proteins. And this is a term you will hear, that people are on protease inhibitors. People who are on protease inhibitors are stopping this process here. And it is a very, very successful therapeutic approach. Okay. Now, the reason that in our COVID epidemic that is interesting is because it suggests to us that there may be ways in which we could interfere with COVID as well. And these are in fact being tried. You may have heard of a drug called remdesivir, which is being used with some success, not outstanding success, but with some success. Remdesivir interferes with the production of new copies of the viral genome. And then there is work being done looking at inhibition of the proteases, which would produce the proteins, the viral proteins of COVID as well. Okay, so just a quick word about the vaccines, and especially about vaccines that are being used in COVID, in the current COVID epidemic. In general, a vaccine is a harm that's derivative of usually either of a pathogen or of something very like the pathogen, which we use to educate the immune system. The immune system is able to do two things. First of all, respond immediately to an infection and produce antibodies which limit that infection. But the second more important function of the immune system is that the immune system can form memory. It can form memory of previous infections. And if ever it sees something similar, it immediately leaps into action to prevent the infection from proceeding. Now, this is used extremely successfully and has been used extremely successfully with minimal risk in many, many, many diseases. The most outstanding of which, of course, is smallpox. Smallpox no longer exists as a viral disease on the planet because of the worldwide usage of a very good smallpox vaccine. Most childhood diseases now can be prevented by the use of vaccines such as the measles, mumps, rubella vaccine, chickenpox vaccines, etc., etc. The importance of vaccines is that there are very, very few drugs which will operate directly to interfere with a viral infection, which will operate to stop a viral activity. There are a few, as for instance, I just told you for HIV, and some that are being developed for COVID. But these drugs usually, even if they slow down the progression of illness spectacularly, as they do with HIV, they don't really offer a cure. If somebody who's been on long-term treatment for HIV stops their treatment, they very quickly revert to a disease status. So the vaccines are extremely important. The COVID vaccine, there's one set which are old-fashioned vaccines where the vaccine is developed by modifying those spike proteins and injecting the spike proteins directly so that the body recognizes them as foreign protein and then develops a reaction to them. But the more effective vaccines in COVID are actually mRNA vaccines. And what they are doing is we are introducing a small piece of messenger RNA into the host cells. And that messenger RNA is translated by the host machinery to produce a viral protein, the spike protein. And just on its own, it cannot do anything. It can't cause disease or anything, and it is actually expressed by the host cells. It's released and is recognized by the immune system, which reacts against it and develops both antibodies and then develops memory as well. And these vaccines have proved to be extremely, extremely effective. Vaccines always offer two things. They may prevent illness. They don't always prevent illness entirely, but if they can sometimes prevent any infection from developing to illness. And if illness develops, they modify the illness so that it is much less severe. The onset of the body's immune responses are much more rapid. So the illness is shortened and is modified. If you want to learn more, please go and check out on the CDC webpage and read about vaccines. This is especially important because you're going to be asked by people who know that you're doing biology, whether you know about these. Okay, I'm sorry that I had to rush through viruses at such an incredible lick. It's a fascinating topic. But we need to move on to our discussion about the prokaryotes. And these are the two groups. Remember, I showed you before that their relationship is not as close as one might think, but they are both prokaryote groups. They are single-celled organisms. If they associate with one another, their association is a very loose association. It's not truly colonial. They just, sometimes cells divide and remain associated with one another, or they may form biofilms, but they are essentially single-celled. And these are two great domains, bacteria and archaea. The two domains are in themselves extremely diverse, and they occupy virtually every single habitat on the planet where life is possible. They have even been discovered in rock miles deep in mines, for instance, in South Africa. They have been extracted from rock miles deep, and they have been found in hot water. They're found in marine systems, freshwater systems, in the soil, etc., etc., etc. If life is possible, you can be sure that there are either bacteria or archaea are there. They were, of course, the very first things to evolve on our planet, and their ability to survive in these extraordinary environments is a reflection of their history on the planet. Their population size is absolutely enormous. They are the most abundant organisms on the planet. And there are various things that have allowed them to be so incredibly successful. The first thing is they reproduce very, very rapidly. They're small. They have a small genome, and they can grow to the size which is sufficient for them to divide very, very rapidly. Cells, for example, like Escherichia coli, which is the standard kind of laboratory prokaryote, in a good rich medium at a good temperature, like 37 degrees Celsius, can go through its entire life cycle, reproducing its entire genome, dividing in less than 20 minutes. So you'll realize that their growth is exponential. As long as they are provided with enough nutrients and conditions are correct, their growth, they double up every 20 minutes, right? It depends. There are other bacteria which grow much more slowly, but whatever their generation time, each generation they're doubling. So they can reach enormous numbers very, very quickly, which is a huge advantage because it means that a very few cells entering a suitable environment can very quickly monopolize that environment. The second thing that's important about them is that the prokaryotes are able to mutate very rapidly. And as a result, they can develop enormous diversity very, very quickly. They can adapt to changing environmental conditions very quickly. Their evolution is extremely rapid. We can drive prokaryote evolution experimentally in a matter of days. We can drive evolutionary of antibiotic resistance, for example, experimentally, extremely rapidly. In addition, they have many different adaptations, many extraordinary adaptations. They are adapted, for example, there are bacteria which are adapted to living in boiling water. They have specialized proteins which protect specialized nucleic acid structures, which enable them to survive the temperatures that high. Others, for example, here, for example, this is a bacterium which is able to produce a spore. This is called an endospore. It's not a reproductive structure, it's a resting structure. It's a structure produced when environmental conditions become harsh, when the cell itself, which is called the vegetative cell, can no longer survive. As it's approaching that point, it forms this specialized cell with a thick cell wall, which is highly resistant to high temperatures, to dehydration. You see, they're even resistant to disinfectants and all sorts of things. There's hardly anything that can penetrate this thick cell wall. They can remain dormant. They have virtually no metabolism. They can remain dormant for thousands of years until the correct environmental conditions develop again, and then they germinate, and then they go off immediately, start reproducing. So there are many other adaptations in the prokaryotes, but they are extremely diverse in their biochemistry, their metabolism, and their adaptations. Remember also that all of the major biochemical metabolic processes, photosynthesis, cellular respiration, all of these processes evolved in the prokaryotes. And when we look at the current day prokaryotes, extant prokaryotes, we see those processes almost like a fossil record of our own metabolic processes. So here are some of the different forms. These spherical ones are referred to as being cocci, C-O-C-C-I. Rod-shaped ones are referred to as being bacilli. And then there are spiral bacteria as well. There are two forms of spiral. This is just an ordinary, this is a spiral bacterium where the whole body is spiraled like this. But there are also bacteria which form a tight corkscrew shape, which is wrapped around a flagellum. And those are the spirochetes. So there are many different morphological forms, and they also group together differently. Sometimes they are individual, sometimes in twos, sometimes in large not colonies, but in large chains of cells which are loosely joined together. The bacteria, not Archaea, but the bacteria have on the outside of their cell membrane, they have a thick cell, they have a cell wall. Forget the thick part. They have a cell wall, a very strong cell wall. Now that cell wall is made of a polysaccharide called peptidoglycan. And if you look at it under the electron microscope, what you see it is like a net. So it has no ability to regulate the passage of molecules the way that the cell membrane does. It forms a very strong net, a mesh around the outside. It gives the cell its shape. It preserves the shape of the cell. It is dynamic, it's broken down and built up, broken down and built up the whole time, especially as the cell grows. But its main function is as an osmoregulatory mechanism, because these prokaryote cells suffer the problem that water continually floods into them because the cytoplasm is more concentrated than the outside. So the water from the outside is continually pouring into the cell by osmosis, and the cells tend to swell. And the cell wall provides a mechanism whereby the cell membrane swells up against the cell wall, and the cell wall prevents it from bursting. So it is a protective mechanism. Some bacteria have a very thick cell wall. Others have a thin cell wall. And you may have heard that there are two particular groups in the bacteria. They're not the only two groups, but there are two particular groups in the bacteria which we recognize. And these are the gram-positive bacteria and the gram-negative bacteria. Gram-positive bacteria have a great big thick cell wall around them. And they retain stain during a process called the Gram stain. Gram-negative bacteria only have a thin layer of peptidoglycan, and they have another membrane which looks very similar to the cell membrane. It's around the outside, and those are Gram-negative bacteria. Just need to do something because my my earphone is chirping at me. Just give me 1 second. Okay, I hope that works. I've got wires dangling off me now, but that's the best I can do. Okay, so those are the bacterial cells. Archaea do not have peptidoglycan in the cell wall. They have a variety of other polysaccharides, but they still have to have this kind of protective structure, but they don't have peptidoglycan. Remember that the only other groups that share this cell wall feature, and for much the same reason, by the way, are the plants. So the plants, the cell wall is composed of cellulose. Fungi also have a cell wall, and their cell wall is composed of chitin. So in each of these radically different groups, they're having a similar structure, the cell wall, but derived from different sources. Okay, so the prokaryotes, obviously, since they do not have any internal mechanisms, any internal membranes, all of their metabolic function, everything is scattered throughout the cytoplasm. And in the prokaryotes, the genome consists of a single DNA molecule in a circle. So unlike the eukaryotes, which have linear chromosomes, the prokaryotes have circular chromosome. And that chromosome can be very long. It's large. And even though it's not membrane bound in the cytoplasm, it doesn't just lie loose and random. It's very carefully organized, and it has to be carefully organized because it has to be easily accessed. The genes need to be easily accessed. The cell basically has to know where things are. So it is very carefully organized. There are some proteins associated with its organization, but they're unlike the I'm sorry. I have to do this again. Oh, goodness sake.
Gosh, now I think I've lost you now. Oh, I'm just going to end.